Greetings, I'm Sowmya and I'll be presenting a poster titled Understanding Flux Rerouting in Metabolic Networks Through an Analysis of Synthetic Lethal Pairs. So I'll briefly go over the introduction, I'll motivate why we're doing this, and also enumerate the questions that we seek to address. So the main motivation is we wanted to study robustness in detail. Robustness is an essential attribute to all organisms, and what it does is it ensures viability in the face of perturbations, and how it achieves it is it introduces redundancies in the networks. And it does this by using alternate pathways, and an easy way to look at alternate pathways is to study higher order synthetic lethals. And the higher order synthetic lethals can vary in their degree of complexity. Some can be just an after effect of gene duplication, or some could be reactions that are present in different submodules, metabolic submodules, which still compensate for each other. And in particular, in the study, we focus more on double lethals, synthetic double lethals, and they'll be referred to as DLs for the rest of the talk. There are certain previous studies which also looked at synthetic double lethals, and what they did was they classified these DL pairs into two categories, that is the PSL and RSL, classic synthetic lethals and redundant synthetic lethals. These two classes vary on just one point, that is the activity of the reactions, selective activity of the reactions that comprise a double lethal. So in PSL, only one reaction is active. The second reaction becomes active only when the first reaction is inhibited. And in redundant synthetic lethals, both of these reactions are simultaneously active in order to ensure, say, maximum biomass flux or something. If one of these reactions is deleted, the fluxes are rerouted in such a manner that the organism is still viable and the biomass is still high. So with these points that are presented over here, we came with a set of questions. And the first one was, how the reactions that are present in different metabolic submodules even compensate for each other? Can we understand the complex flux reroutings that underlie synthetic double lethals? Can we, if possible, identify the minimal set of reactions through which fluxes need to be rerouted? And regarding the PSL and RSL pairs, what we wanted to see was, what is the metabolic efficiency of the inactive reactions versus the active reactions? And what kind of reactions make up the RSL pairs, especially since both the reactions are simultaneously active? So to answer these questions, we took a twofold approach. So we proposed an algorithm called min rerouting, which answers the first three questions. And then we used an approach which uses conditional FEA to answer this last two questions. So flux rerouting, what do I mean by flux rerouting in a network? Take this sample network as an example. There are 7 metabolites, 8 reactions. We know the metabolites, edges are reactions, and there are 2 exchange reactions. A and B are the input metabolites and things like that. There are multiple double lethals. We take R4 and R6 into consideration. Say if R4 is inhibited, then R6 is active. And if R6 is inhibited, then R4 and R5 becomes active. So when R4 is active, an exclusive set of reactions which become active is R5. And when R6 is active, the exclusive set of reactions which become active is just R6. So if we take the rerouting set and make it exclusive, we'll have 4 R4, R4, and R5, and 4 R6, R6. And say now that it's just not these reaction fluxes which vary, the fluxes through any of these reactions could differ. And say that the flux through reaction R3 alone differs significantly. So we would consider R3 as also one of the reactions which comprise the whole rerouting set for the reaction pair R4 and R6. So when we say a common rerouting set, that would refer solely to R3, and that the size of the common rerouting set in this example would be 1, and the size of the total rerouting set would be 4. It will comprise of R3, R4, R6, and R5. Okay. Now, since we want to use an algorithm to identify this minimal set of reactions, this is just a very simple example model, but it gets much more complicated when we take an actual genome scale metabolic model. So what we wanted to do was we had a set of wish list of sorts for our algorithm to follow. And what we wanted to do was simultaneously solve for the flux distribution that satisfies all the stoichiometric conditions and maximizes the biomass constraint, and also minimizes the number of reactions with varying metabolic fluxes. This was our wish list. And we named our algorithm min rerouting. It's a constraint-based optimization approach. And the mathematical formulation is as follows. This is a generalized phenom expression. What we do is we apply a MoMA-like optimization to minimize the phenom of the difference of the flux between V delta R1 and V delta R2, and subject to all these stoichiometric conditions. And once we obtain these two flux vectors which are close to each other, phenom close to each other, what we do is we calculate the number of reactions which have different fluxes. We identify the minimal rerouting set, the size of the minimal rerouting set, size of the common rerouting set, and even the total flux difference between these two flux vectors. The results that we obtain are as follows. We apply the algorithm for a total of 11 different organisms, and this is the results that's shown over here is for Klebsiella pneumoniae. And one thing that we've identified was zero norm optimization. As expected, identify the minimal rerouting set, followed by 1 norm and 2 norm. And 1 2 norm solutions essentially give the same range of common rerouting set and even SO cluster set, the min rerouting cluster set. And one interesting thing is when we only consider the reactions with significant flux change, we see that the 2 norm flux difference is lesser than 1 norm followed by 0 norm. We would generally expect it to have like 1 norm as the least, but when you consider a significance for the flux difference, it turns out 2 norm has a much lesser flux difference. In order to address the PSL RSL activity question, what we do was we applied conditional FEA, as I mentioned before. And the main idea behind the conditional FEA is to identify whether both the reactions can simultaneously carry fluxes, or whether one reaction can carry a zero flux when the other reaction is active. The previous study, which I mentioned, actually uses an FBA-based approach, and our approach using a conditional FEA improves on it. How it does is when it's an FBA solution, it's just one instance of all the possible solutions that's available. And when we use an FEA, it becomes more rigorous. So what we want with this FEA is we identify the minimum and maximum fluxes that can be carried by a single reaction, and then we constrain and see whether both the reactions are simultaneously active or not. If they're not, if they both cannot carry, if there is a chance that one of them carries a zero flux, which means that the product of the signs of the flux, meaning max flux, will be less than zero. We constrain one of the reaction to be always active and see whether the other reaction can carry a zero flux. So if the reaction is active and the other reaction can carry a zero flux, it means that it falls under PSL. If it cannot carry a zero flux, then it means that it falls under an RS. And the results that we obtained for this part of study was followed by a PFBA, which was applied on all the reactions to identify the reaction flux. And PFBA was used to identify how metabolically efficient each of these reactions were. And what we found out was that about 80% of the models had greater than 90% of the RSL reactions to belong to a PFBA optimal pair, which meant that because both RSL reactions are simultaneously active, it meant that the PFBA optimal are the more metabolically efficient method to see this. And this is the results that was obtained for E. coli core. E. coli core, as we all know, is the core model of the E. coli. And we see that 100% of all the RSL reactions actually belong to PFBA optimal pair. And to the discussions, we see that min rerouting can be used to understand the complex metabolic reroutings that generally occur, and especially it can be used to understand the reroutings that occur in case of disease conditions. And this could be used in order to understand more about cancer, and it's actually being used by another colleague of mine to understand how the rerouting occurs in cancer. And it could go a long way in helping us find better therapeutic cures too. Thank you.